Hi, my name is Itai Yanai, and I'm from the Faculty of Biology at the Technion, Israel Institute of Technology. In a new study, now published in Developmental Cell, members of my lab and I have done an analysis that provides evidence for the discontinuous nature of embryonic development at the molecular level. Since the time of Aristotle, biologists have looked at the embryo and have seen what appears to be distinct stages as the embryo proceeds from egg to adult. Now, the appearance of such stages, such milestones, may reflect the modularity inherent in development, or they may just be an interesting artifact of a fundamentally continuous process. What we sought to do in this study was to then ask what is really the relationship between the external morphology of the embryo and the underlying molecular activity. As a molecular proxy for gene regulatory states, we used mRNA levels. We invoked techniques that allow us to measure the gene expression levels of all the genes simultaneously, therefore providing a global picture of what the embryo is doing throughout development. This is Michal Levin. She is a graduate student in my lab and the lead author of this work.
Hi. When observing C. elegans nematode embryos, I identified 10 developmental stages spanning the whole of embryogenesis. Interestingly, other species go through these same stages, but at different rates. The idea was that we could use the different developmental timings to ask if there is a molecular basis to developmental milestones. To do this, I collected embryos from each of the species at the different stages and assayed their transcriptomes.
Our hypothesis then was that if milestones exist, then the transcriptomic relationships between them should be unaffected even despite species-specific timing variations.
As the first clue that development is actually punctuated as opposed to a steady-state process, we saw that certain stages are followed by a high acceleration of the number of genes upregulated per minute.
Intuitively, what we would expect is that the more time between a pair of stages in the embryo, the more gene expression differences we should find. And this is exactly what we observe. Consider, for example, stages 3 and 4. There are differences in timings between them in different species, and indeed, this is correlated with the gene expression differences that we see. However, for one pair of stages, we did not see this pattern. We saw that no matter how much time there is between them across the different species, it does not matter at the gene expression level. That is the same. It's as though the developmental process occurring at these stages is punctuating the gene expression program.
We show evidence that one of these stages might actually be the nematode phylotypic stage, a stage at which animals of the same phyla are morphologically very similar. We saw that genes coming up at this stage are highly enriched for developmental regulators like home-box transcription factors and signaling pathways. This set of genes is also enriched for knockdown phenotypes like paralysis, indicating that this stage is actually really important.
Comparing across other data sets, we saw that the same genes that come on during the nematode phylotypic stage are generally the same as those that come on during the phylotypic stage of frogs and flies. It's as though this is a strongly conserved evolutionary stage in all bilaterian animals.
To sum it up, our analysis revealed that there is a molecular basis for certain stages we call milestones, whose transcriptomes are invariant to time differences between species, and in a sense punctuate gene regulation in the embryo.
Our results suggest that development across closely related species might evolve by timing changes between milestones. However, it's tempting to speculate that across large-scale evolutionary timescales, it is the milestones themselves that diversify.